So from today onwards, let us start with the session on water-soluble vitamins, and the first one being vitamin C, also called as the ascorbic acid. So let's move on to see what is the actual structure of vitamin C or the ascorbic acid. This is the basic structure of vitamin C or ascorbic acid. This is how an ascorbic acid looks like structurally. Moving on to the chemistry of vitamin C. First of all, the ascorbic acid is an enediol lactone of an acid with a configuration similar to that of sugar L-glucose. It is comparatively a strong acid, stronger than acetic acid. This is the thing we need to keep in mind, that ascorbic acid is strong acid as compared to acetic acid. Naturally occurring vitamin C is L-ascorbic acid. It is stable in the solid form and in acidic solutions, but is rapidly destroyed in alkaline solutions. Let's see how this is synthesized. First of all, it is not synthesized in humans. Some of the lower mammals like rats can synthesize the vitamin, that is vitamin C, from glucose by the uronic acid pathway. Man, monkey and guinea pigs lack the enzymes necessary for the synthesis of vitamin C. They cannot convert gulonol acetone to ascorbic acid. Hence, the entire human requirement must consequently be supplied by the diet. So, we need vitamin C to be supplied to us through our diet. We cannot synthesize this vitamin C. Absorption, distribution and excretion. First of all, vitamin C is readily absorbed from the small intestine, peritoneum and subcutaneous tissues. It is widely distributed throughout the body. Some tissues contain high concentrations as compared to the others. Local concentration roughly parallels the metabolic activity, found in descending order as follows: pituitary gland, adrenal cortex, corpus luteum, liver, brain, gonads, thymus, spleen, kidney, heart, skeletal muscle, etc. From maternal blood, it can cross the placental barrier and it will be supplied to the fetus. So, vitamin C can be supplied to the fetus as it can cross the placental barrier. The normal human blood plasma, it contains approximately 0.6 to 1.5 milligram of ascorbic acid per 100 ml. The vitamin exists in the body largely in the reduced form with reversible equilibrium with a relatively small amount of dehydroascorbic acid, which is the oxidized form. So there is oxidation which results into the formation of this dehydroascorbic acid. But vitamin C exists in the body in the reduced form. Occurrence and food sources. It is widely distributed in plants and animals. In animal tissues, there is no storage for vitamin C and it contains small amount, but highest concentration in metabolically highly active organs such as the liver. As we all know, liver is metabolically active organ. Other organs includes adrenal cortex and corpus luteum. So these are the places where the concentration of vitamin C can be seen to be raised up a bit as compared to other organs, but there is overall no storage into the animal tissues. Dietary sources. These are chiefly vegetable sources. So mostly vegetables are the main suppliers of vitamin C. Good sources are citrus fruits, example orange, lemon, lime, etc. Other fruits like papaya, pineapple, banana, strawberries are also helpful in supplying vitamin C. Amongst vegetables, leafy vegetables like cabbage and cauliflower, germinating seeds, green peas and beans, potatoes and tomatoes are also good sources of vitamin C. Amla, as we all know, and we need to keep this in mind, is the richest source of vitamin C. Considerable amount of vitamin C activity is lost during cooking, processing and storage because of its water solubility. Since vitamin C is water soluble, the activity of vitamin C is lost because of this processing activities, and its irreversible oxidative degeneration to inactive compounds. That is the other reason for its activity being lost during cooking, processing and storage. So the first reason being it water soluble, and the other one is its irreversible oxidative degeneration to inactive compounds. Let's look at the functions of vitamin C. First of all, it plays a major role in collagen synthesis. Hydroxyproline and hydroxylysine are important constituents of mature collagen fibers. Pre-collagen molecules contain the amino acids proline and lysine. They are hydrolyzed by corresponding hydroxylases in the presence of vitamin C, Fe2 plus and molecular oxygen. In scurvy, failure of conversion of pro-collagen to collagen due to the failure of hydroxylation may lead to rapid destruction of the collagen intermediates. So in cases of scurvy, if there is failure of conversion of procollagen to collagen due to the failure of hydroxylation, it may lead to rapid destruction of the collagen intermediates. This is the way it has been shown, the conversion of proline to hydroxyproline and lysine to hydroxylysine. And vitamin C plays a major role in this. Moving on to the next function, ascorbic acid is required for functional activities of fibroblasts and osteoblasts, and consequently for the formation of MPS of connective tissue, osteotissue, dentine and intercellular cement substance of capillaries. Absorption of iron is the major role played by vitamin C. Ascorbic acid in food helps in the absorption of iron by converting inorganic ferric ion to the ferrous form. Also by forming water soluble ferrous ascorbate या फिर iron ascorbate chelate. This is the important water soluble compound which is formed. Also helps in mobilization of Fe from its storage from ferritin. Disturbances of these functions may contribute to the development of hypochromic microcytic anemia in scurvy. Absorption of Fe both in normal or Fe deficient patients is increased by over 10% after administration of vitamin C. So that is the end to the topic on vitamin C. Next we shall look at water soluble vitamins, which includes the vitamin B complex. Thank you very much and stay tuned for more such videos.